My name is Sam. Let me see. How do I advance? Oh, yes, there we go. My name is Sam Booth. I'm a PhD candidate at the University of Manitoba at the Rady Faculty of Health Science. I'm working in Dr. Ji Hun Ko's laboratory. So we're a translational brain imaging laboratory, but we also have done some work in animal models in the past, studying neurodegeneration and mood disorders and all kinds of different stuff. So yeah, I'm a PhD candidate. I'm actually defending in a few weeks, so this is a good opportunity for me to practice in front of everybody. So any feedback or questions is definitely welcome. Okay, so I'll just briefly introduce the disease model that most of my work is in, which is Parkinson's disease. So this is the second most common neurodegenerative disorder after Alzheimer's disease. And the incidence has actually doubled since the 1990, and it's expected to continue to increase, perhaps even doubling by 2040. And this is mostly due to almost every country in the world has a rapidly aging population. And so finding better ways to manage this rapidly aging population that's at risk for Parkinson's disease is a big public health concern and a big policy concern. And so this is kind of what has motivated my research. So for those who may not know, Parkinson's disease is a progressing neurodegenerative disease. The most well-known symptoms and the basis for the diagnosis of Parkinson's disease is typically these motor symptoms, bradykinesia, resting tremor, rigidity, and in the more advanced stages, patients develop postural instability, they have gait disturbance, they have freezing and a high risk of falling. But in addition, Parkinson's disease patients also have a host of non-motor symptoms, and it's increasingly recognized that during this prodromal phase, there's actually a lot of subtle non-motor symptoms that are actually the earliest stages of Parkinson's disease, such as sleep behavior disorder, mood disorder, and there may be subtle dysexecutive disorder actually starting in the early stages. And of course, in the advanced stages, patients often develop worse reaction to the Parkinson's medication, these drug-induced complications, which I'll be discussing in greater detail later on. So Parkinson's disease is a very complex pathophysiology, but one of the biggest hallmarks of Parkinson's disease is the degeneration of this nigrostriatal dopamine pathway. So these are dopamine-producing cells that project from the substantia nigra in the midbrain to the striatum, and there they regulate these corticobasal thalamic cortical loops. So those regulate voluntary movement. They also regulate attention, executive function, a whole bunch of different things. So for whatever reason in Parkinson's disease, these cells seem to be selectively vulnerable and die. And this causes the classical Parkinson's disease symptoms. About 90% of cases of Parkinson's disease are idiopathic, meaning we don't know what the root cause is. So kind of the theme of my PhD research is trying to study in more detail these things that limit our effective treatment and management for Parkinson's disease. So these include the development of these drug-induced motor fluctuations called Levodopa Induced Dyskinesia, which I will talk about in more detail later. And additionally, cognitive impairment in Parkinson's disease, which in many cases can cause worse disability than the actual motor symptoms of Parkinson's disease. And actually has a big effect on caregivers and the patient. And these are actually very much more difficult to treat than motor symptoms of Parkinson's disease. So more information needs to be learned about this. So the gold standard treatment for Parkinson's disease is levodopa. It's been that way since the 1960s, and it very effectively manages motor symptoms, especially in the early stages of the disease. There's a lot of other supplementary treatments for Parkinson's disease and some like deep brain stimulation and stuff, which are used more in a narrower patient population. But still, like, 80 years later, levodopa is the big drug for Parkinson's disease. And it's basically it's a form of it's a precursor to dopamine that can actually cross the blood-brain barrier. And once in the brain, it gets rapidly converted to dopamine. And so this, extra dopamine in the brain is what robustly alleviates Parkinsonian symptoms. However, there's an issue with levodopa, and that is that as the disease progresses, as time goes on, the patients develop, first of all, a lengthening of the off period. The drug works for a shorter duration, and the patients develop these things called dyskinesias, Levodopa Induced Dyskinesia, the most common being peak dose dyskinesia. And these are abnormal hyperkinetic movements that the patient develops when they're at their peak plasma concentration of levodopa. And the reason this is a big barrier to effective treatment is because it means they're spending time where the drug should be helping them manage the Parkinsonian symptoms the most. They're developing these dyskinesia symptoms, and this requires a lot of careful work and balancing with the clinician to try and get their medication to a point where they can minimize these dyskinesia symptoms and actually lengthen the therapeutic window. So the cause of levodopa-induced dyskinesia is kind of its own kind of subfield within Parkinson's disease research. And there's a lot of different potential mechanisms at play, as you can see. And this likely involves a combination of presynaptic and postsynaptic alterations. So by presynaptic, I mean that there's a change in how levodopa is converted to dopamine and released in the striatum. That's dysregulated in Parkinson's disease patients. And then there's also a whole bunch of postsynaptic and network changes that occur downstream of that, starting with the medium spiny neurons in the striatum, which actually receive dopaminergic input. So one of the first projects I was involved in at the lab when we started was this project. And so there's some evidence that in levodopa-induced dyskinesia, the vasomotor response is messed up to the point that there's actually a dissociation between blood flow and metabolism. And so we performed this small study using perfusion, MRI, and FDG PET. So FDG PET measures the metabolism in the brain. It's basically a radio-labeled glucose analog that gets taken up. And what we found is that we were able to differentiate a lot of lid and non-lid patients by measuring the dissociation between cerebral blood flow and cerebral metabolism in the striatum when the patient has levodopa. And this is something that's been seen in also many other studies as well. So we wanted to, in my first paper in the lab, we wanted to investigate this in more detail using our animal model. So we investigated angiogenesis and endothelial vasomotor response to dopamine treatment in a rat model of Parkinson's disease. And our aim was to see which of these are most relevant in differentiating the dyskinetic and non-dyskinetic animals. So we use a lesioned rat model of Parkinson's disease. So essentially, we inject a 6-OHDA, which is a toxin that selectively kills these dopaminergic cells into the brain, and this induces Parkinsonism in the animals. And then the animals are given daily levodopa treatment for 22 days. And during this period, the animals develop levodopa-induced dyskinesia. They develop these abnormal involuntary movements when we give after about 20 minutes after their levodopa dose. And we measured these at 3 time points using this abnormal involuntary movements test. It's basically a very simple observational test measuring the severity of dyskinesia symptoms in the animals. And another thing that we wanted to do is what we found and what we found from the literature is that most studies give animals a relatively high dose, 10 milligrams per kilogram of levodopa. And what we found is that this induces dyskinesia symptoms in all the animals from day one. And that basically 100% of the animals develop dyskinesia. So we had a kind of a dose finding study where we wanted to reduce the dose of levodopa in order to create a progressive model that might be suitable for longitudinal studies. And additionally, this one means that some animals develop dyskinesia and some animals don't. And what we found is that the lid and non-lid animals all had similar lesion size, so this effect couldn't be chalked up to incomplete lesion size.
I just had a couple of questions. One
Gotcha.
was, you've been saying animals. These are mice?
These are rats.
They're rats.
Yes.
Because, so I forget which way it goes, but this doesn't actually work in one.
Yeah, there's the mice don't really develop levodopa induced dyskinesia, which is why we had to use a rat model. Yeah, we have all female animals, and this is a bit of a stupid reason, but it's because they fit better in the bed in the scanner. They don't get so big. We don't really have the sample size to really look at sex differences, but yeah, there is actually in levodopa induced dyskinesia. It is actually more common in females compared to males. They are quite young, I think around 3 to 5 weeks. Yeah, they're still quite young, yes, but they recover better from the lesion when you inject them young. Yeah, we screen them with a cylinder test, which basically it's a hemi lesion model. So there's a complete lesion in one side of the brain, and in the other side of the brain is still intact. And so we can measure the forelimb asymmetry using this test called the cylinder test. The cylinder test isn't super specific, so we also harvest the tissue and do immunostaining for the for tyrosine hydroxylase, which identifies the dopamine producing cells, which I'm showing here in the micrograph. Yeah, with greater disease severity, there's greater incidence of dyskinesia, and actually what's been shown in both humans and in animal models that there's actually increased dopamine flux in the dyskinetic patients. So more of it is getting converted to dopamine faster. The best theory for how that is serotonergic hypothesis, and this is because there's serotonergic projections in the striatum as well. And the idea is that in levodopa-induced dyskinesia, these serotonergic neurons are actually taking levodopa and they're actually converting and releasing dopamine. And because they don't express the D2 autoreceptor, that's why they're actually releasing it at non-physiological levels. And there's a huge amount of data actually to support that. It hasn't actually translated into like an effective treatment, mostly because targeting serotonin is often counterproductive for Parkinson's disease. But so I'll talk about this briefly because this is cerebral imaging lecture. But what we were interested in this vasomotor response, and so what we wanted to know if there was angiogenesis, because that had been found previously in like 2006 in some study, but again they used 25 milligrams per kilogram of levodopa, and so we wanted to see if angiogenesis is significantly different between lid and non-lid animals. And so what we found is we actually these micrographs are staining for rat endothelial cell antigen, which stains the vessels, and the red are proliferating nuclei because we gave the animals BRDU, which is basically it's a it gets incorporated into the DNA, so you can then immunostain for the proliferating nuclei. And what we found is that we replicated the findings of previous studies, showing that there was angiogenesis significantly increased in the lesioned hemisphere compared to the non-lesioned hemisphere in the lid animals when we were using a high dose. However, when we were comparing the low dose, the non-lid and lid animals, there actually wasn't evidence of angiogenesis. So we also did brain slice imaging to basically look at the vessels, the microvasculature, and see if there's a difference when we apply dopamine in a bath, whether there's a difference, because dopamine is vasoactive. It does modulate the microvasculature directly. And the thing that's interesting is similar to its effects on neurons, it actually seems to be a bimodal effect where it can induce either constriction or dilation. And what we found is that in our lid animals, it was sharply shifted towards vasodilation. So in the dyskinetic animals in their striatum, when there's dopamine in there, there's vasodilation, increased blood flow. Whereas in the non-lid animals, in the PD animals without L-Dopa, and in the sham control animals, they're all pretty much the same, and it's fairly balanced. Yes, so this study basically, this study was kind of one of the motivations behind it was getting this lower dose model with progressive symptoms so that we could investigate longitudinally. And we did this in a longitudinal brain imaging study where we looked at changes in cerebral blood flow and metabolic rate in an animal model longitudinally. So this is a similar experimental setup to the first study. However, in this study, we're doing PET MR scanning sessions, both at baseline at the first dose of levodopa, after 21 days of treatment off levodopa, and after 21 days of treatment on levodopa. And again, we use these Ames tests to quantify the dyskinesia symptoms. And again, we found that in the lid animals, there's progressive symptom onset of dyskinesia symptoms. In the non-lid animals, there's not. And at the peak of dyskinesia symptoms occurs about 60 to 80 minutes after. Okay. And again, we had similar lesion size between the lid and non-lid groups. No significant difference between the lesioned and non-lesioned hemisphere. This one down here is a sham lesion. Okay. So in order to measure the blood flow, we used a technique called dynamic susceptibility contrast MRI. So with this technique, you inject a bolus of contrast agent intravenously, and you image the passage of the contrast agent, and then you can generate hemodynamic parameters from that. We got structural MRI for spatial normalization purposes. So the animals were given levodopa at minute 0, so that during the DSC and FDG PET scans, that would be where the peak of dyskinesia symptoms would be. Yeah, and the animals were given FDG, which is a radio-labeled glucose analog, so we can look at the metabolic rate in the brain between these animals. Okay, and then from the time attenuation curve of the contrast agent moving through the brain, you can generate hemodynamic parameters. So we generated the relative cerebral blood flow, which was normalized to the whole brain mean, and the FDG standard uptake ratio. And we use a univariate general linear model analysis to try and look at an interaction effect between group, so the dyskinetic and the non-dyskinetic animals, and the drug condition off and on levodopa, because they were scanned off and on consecutive days. And basically what we found is we identified these clusters in kind of the lesioned dorsolateral striatum, where there's a significant interaction effect between the off-to and on-to condition and dyskinesia status. And this area is topographically associated with the corticostriatal motor circuits. So this area kind of made sense to us as a region for further analysis, for post-hoc analysis. So from this cluster, we then extracted the mean FDG standard uptake ratio in both the off and on condition at the first levodopa treatment and after 22 days of treatment. So I'll just show here, yellow is the non-lid animals, and there's a decrease in FDG uptake in this region, or reduced metabolism in this region on levodopa. And this is true from the first dose and after 22 days of treatment. Now, what's interesting is in the lid animals, we didn't see that. It was pretty much the same. And after 22 days of treatment, after the dyskinesia symptoms had progressed, what we actually found was that there's increased FDG uptake within this region. And so when we looked at the literature, what we found is that these regions, particularly the globus pallidus and the putamen, there's evidence that in Parkinson's disease, these regions are tonically hypermetabolic. And we have data, there's data from this in using neuroimaging techniques. And in animal models, there's also more direct measurements of changes in firing rate and stuff like that. And what's interesting is that the ability for reduction in the expression of this metabolic pattern is correlated to the efficacy of treatment. So this pattern is reduced with levodopa treatment. This pattern is also reduced in non-dopaminergic treatments, such as deep brain stimulation. So our data support this, that levodopa actually normalizes or reduces the hypermetabolism in this striatal region. And there's a whole bunch of research that's been performed on the striatum and how there's maladaptive plasticity in the neurons of the striatum, both as a result of Parkinson's disease, but then also as a result of the actual chronic levodopa treatment. These include, there's actually increased glutamatergic input into the striatum in levodopa-induced in Parkinson's disease, rather. There's all kinds of changes in the expression and subtype distribution of glutamate and dopamine receptors. And there's structural remodeling of the neurons, changes in excitability and firing rate. And so what we found that was interesting was that the dyskinetic animals showed a failure to normalize striatal metabolism. And what was interesting is this was found even in the, even in the drug-naive state. So even from the first dose of levodopa, they don't respond the same way that non-lid animals do. And so this indicates to us that there could be a difference in the plasticity of the striatum and the striatal neurons, and that occurs as a result of the lesion or as a result of dopaminergic deafferentiation. And there might be a difference between the animals who later developed levodopa-induced dyskinesia and the animals that remained non-lid. We also did find increased striatal blood flow only in the lid animals in this cluster, and only after the 22 days of treatment. It wasn't present from the first dose. So this shows us that this vasomotor effect that we saw in the previous study probably occurs later on. It's not present before the dyskinesia symptoms progress. And so just to sum up, what we found in this study was increased levodopa-induced blood flow is apparent after dyskinesia symptoms developed, but not at baseline. In the non-lid animals, levodopa normalizes the striatal metabolic metabolism, both in the drug-naive condition and after 22 days of treatment. And this is how you expect levodopa to work if it's working therapeutically. However, in the lid animals, this was abnormal, and this indicates potential avenues for further research using this kind of rat model, looking at how the striatal plasticity underlying lid symptoms, it might be present in the levodopa naive condition, or how that develops longitudinally. So she's asking if the animals might be non-lid at one time point, but then lid at another. No, that doesn't happen. So with the higher dose model, what we found is all the animals have strong dyskinesia symptoms right from the start. The animals, once they develop this, once they have this adverse drug reaction, the dyskinesia symptoms, it's kind of present like clockwork, really. Yeah, it's retrospectively based on which animals develops the symptoms. Yeah. We check the lesion size, and we specifically, with our model, we inject in a location that is pretty much guaranteed to do a very complete lesion. And we check with immunohistochemistry. So it's not to do with the lesion size. We suspect it's differences in how the brain is coping with the lesion. It's transported into them. It also has an effect on other catecholaminergic neurons, but because of the location that it's injected and the distance that it can actually diffuse without getting broken down in the brain, it's just these ones. I mean, it also, we have a lesion in the VTA as well. It's a very small dose, and so you know what, I don't know if anybody has actually studied whether the animals could behave differently, or whether there is some change from it. I think most of that research is probably in humans, and probably looking at things like cancer risk and stuff like that. My understanding is there's not, but that's definitely something that is worth looking into. There does seem to be differences in these the striatal, the medium spiny neurons that project to the rest of the basal ganglia and are involved in these motor loops. There seems to be a difference in those ones in lid and in non-lid. There's been a whole bunch of studies looking at various electrophysiological, morphological, biochemical alterations in those. It's a complicated question. I mean, with the way that we've interpreted this is, and this requires a lot of more complementary data. But the fact is that after they have dyskinesia symptoms, we do see that the FDG uptake is increased in that region. And so maybe at baseline, the fact that they're not responding the typical way that you would expect levodopa in terms of how it's, say, modulating or perhaps normalizing the metabolism in that region, that could indicate that there's a different kind of plasticity that has occurred in the striatum that sensitizes dyskinesia development. So another aspect of Parkinson's disease, which I study, is cognitive decline in Parkinson's disease. So approximately 80% of Parkinson's disease patients also develop some kind of cognitive impairment over the long term. And clinically, cognitive impairment is defined in kind of two stages. There's mild cognitive impairment, and this is diagnosed based on neuropsychological testing. So if they score 2 standard deviations lower than normal in 2 different cognitive domains, they have mild cognitive impairment. And then PDD, or Parkinson's disease with dementia. This is diagnosed based on neuropsychological tests, but also based on consultation with a doctor on how the cognitive deficit is actually significantly affecting the patient's occupational ability, self-care ability. So it's yeah. And cognitive decline in PD has a very high rate of disability and a really big impact on caregivers. And the prognosis with cognitive symptoms is quite unclear. There's a lot of heterogeneity in the cognitive symptoms that patients develop. Some patients remain stable MCI or fluctuate between MCI and normal condition as you test them. And then there seems to be subgroups of patients that decline rapidly into dementia. And what we're interested in is why some of these patients decline rapidly while others remain relatively stable throughout the disease progression. And so this was our study, which was published in 2022. And we were trying to use neuroimaging to predict cognitive decline at the MCI stage in a small cohort of Parkinson's disease patients. And we used FDG PET scans from PD MCI patients. And we used a supervised learning approach in order to stratify the patients called support vector machine. And so the main population that we used for this study is a South Korean population from Asan Medical Center in South Korea. And these patients were Parkinson's disease patients with clinically confirmed mild cognitive impairment. And these patients were followed from about 3 to 10 years. It's a retrospective study, like a chart review study. And we used a training set of patients. So these were patients that were stable mild cognitive impairment. There were 20 of them. And then we had a group of patients that developed Parkinson's disease dementia. There were 23 of them. And the minimum follow-up time was 3 years. So 3 years to develop dementia. And we used this to train the support vector machine model. This is basically a supervised learning technique where you give it a set of data and it finds a way to optimally separate the two groups. And then you can give it new data and it will assign a group, basically. We had a smaller validation set, holdout validation set of stable MCI and PDD converters. And then we also tested a few different cohorts, such as Parkinson's disease dementia patients, Parkinson's disease normal cognition patients, and normal controls. And again, the FDG PET scans that we used to train the model are ones that were taken when the patient was Parkinson's disease MCI. So not when they had dementia. So these are the baseline scans that we used. We are testing for all of the patients when they're scanned. They have Parkinson's disease with mild cognitive impairments. And it's a retrospective study. So it's the patients who later developed dementia. We more just wanted to test these ones to see what the model classifies them as. Basically like this little set here, because this is not the clinical category that the classifier was trained on, but for qualitative reasons, we wanted to see what the expression of this pattern was in those groups. Let's see here. Okay, so these are the patient characteristics. So between the stable MCI and the MCI to PDD converters, there was similar age and similar disease duration, similar disease severity as well. So these groups were fairly well matched. In the validation set, it was a little bit less of a good match, but it's quite a small holdout validation set because we didn't have a huge amount of data. And so what we also did is we used a permutation testing approach to try and select for the brain regions that are the most robust predictors of cognitive decline. So basically how this works is we have our stable MCI scans, our MCI to PDD converter scans, and we generate the actual support vector machine model. Then we permute the class labels and generate a model based on the permuted class labels. And then what we can do is we can compare the number of times that the magnitude of these predictor coefficients from the models are greater than the predictor coefficients in the real model, the W model. And from that, we can generate a P map or a map of P values from the permutation test to select for these regions that are the most robust predictors of cognitive decline in Parkinson's disease. And we did this so we can get a pattern that we can kind of qualitatively assess which metabolic changes are poor prognostic indicators in Parkinson's disease. So this is the pattern that we generated. What we found, so these are the regions where either increased or decreased FDG uptake was associated with conversion to dementia within an average of 5 years. And so we had increased FDG uptake in prefrontal cortex, anterior cingulate, midbrain, putamen, and globus pallidus, and then reduced FDG uptake in the middle temporal, dorsal prefrontal cortex, precuneus. So what this looked like to me is this is like a kind of a pattern of posterior cortical atrophy was a poor prognostic indicator or an indicator of accelerating cognitive decline, whereas relative preservation in these more frontal and basal ganglia areas was observed. And this fits fairly neatly into a lot of the literature that's been in Parkinson's disease. So as I said, there's a very, very heterogeneous progression and also symptom expression in the cognitive symptoms of Parkinson's disease. And one of the models that's used to explain this is called this dual syndrome hypothesis. So the idea is that most or all Parkinson's disease patients have some kind of dysexecutive, some kind of executive deficit. And this makes sense because it's the dopaminergic cells regulate working memory, attention, executive circuits. Whereas what we found is that there's maybe a subgroup of patients that perform poorly in things like semantic fluency, visuospatial skills, hallucinations, poor verbal memory. Those neuropsychological characteristics are typically very poor prognosis for cognitive outcome in Parkinson's disease. And these may have that more posterior cortical substrate in the posterior parietal regions. And so this fits in a bit with our data where we showed that reduced metabolism or atrophy in these posterior cortical regions was a poor prognostic indicator in our data. So the classifier, so with tenfold cross-validation, we had quite good sensitivity and specificity in differentiating the stable MCI patients from the patients who would later develop dementia. However, it was a little bit worse in the validation set. It was about an average accuracy of 73%, although the sample size is quite small and it's arguably a fairly difficult classification task, stratifying the patients at baseline like that. And so there's actually, at the time that I checked, there's no other neuroimaging Parkinson's disease studies that use this kind of supervised learning to try and predict future cognitive decline. There's a lot of cross-sectional neuroimaging studies between PD-MCI, PDD, and there's a lot of variability in the results, I would say. However, there has been a lot performed in Alzheimer's disease. I read a meta-analysis that included at least 116 papers using PET and MRI. And the accuracy at predicting future Alzheimer's development was in the range of 70 to 80%. So this indicates that it's a difficult classification task. So we're doing okay basically in our external validation set. Yes, of course. Yeah. We didn't include age as like a covariate in the model, but yeah. Okay. Okay. Are we good to move on, or? Okay, cool. Okay. So as I said, we we did test the model out in some different cohorts, just sort of for qualitative reasons. So none of the normal controls were classified as PDD converters, which is what you would expect. And actually very few of the Parkinson's disease patients that were non-demented were classified as PDD converters, even though they had similar disease duration and similar severity. So I think that supported the idea that the classifier is picking up on a cognitive substrate rather than something that just correlates with the severity of Parkinson's disease, because almost all of the clinically confirmed Parkinson's disease dementia patients were classified as PDD converters, which is again what we'd expect. These are idiopathic MCI patients, so this is a little bit inconclusive without having follow-up data on whether they develop dementia or not. Interestingly, the DLB patients that we tested and the Alzheimer's disease patients were both overwhelmingly classified as PDD converters, which shows that it's maybe a similar pattern of atrophy in those groups. Another thing that we did is we had a group of 10 patients with Parkinson's disease. Unfortunately, they weren't really well defined in terms of their cognitive symptoms, but we tested them both off and on levodopa, and we found that there's almost no difference in score depending on levodopa off or on. And what this shows us is that this pattern is not modulated by dopaminergic drugs. So that also supports our idea that this is maybe a non-dopaminergic substrate, like a posterior cortical substrate. Yes. So to close, so Parkinson's disease is the fastest growing neurodegenerative disease. And as I said, levodopa is still kind of a big pillar of Parkinson's disease treatment. And that's what's prompted this field of study, looking at these levodopa-induced motor fluctuations. And additionally, cognitive symptoms in Parkinson's disease. Levodopa almost never helps with any of them. There's also a big gap in actually having good treatments for cognitive decline in Parkinson's disease. And and so neuroimaging and has a potential utility in identifying kind of the neuroanatomical and neurochemical substrates of cognitive decline in Parkinson's disease. And we're hoping that later on this can help with the stratification and subgrouping of patients, because if you can try and select for the patients which are at greater risk for cognitive decline, you may have greater power in actually testing disease-modifying treatments in that group. So that's kind of a direction that we wanted to take this kind of research with. And with that, I will just thank everybody for coming today, and I would be happy to take any more questions. Thank you so much. Thank you.
All right. Thanks a lot, Sam, for the great talk. Do you have questions? We have a few minutes of questions from the audience.
Yes.
Gabe, then Lonnie.
Why an SBM?
We had performed a previous study where we compared a bunch of different techniques. in our Alzheimer's disease population, and SVM was the best performer. So that's why we chose SVM. Philosophically, there might be some things that are desirable about SVM. I think it's like naturally kind of robust to outliers or extreme cases because the model is built basically just using those subjects that are most difficult to differentiate. So that's why we used SVM. Yeah.
Just maybe as a follow-up, how does SVM deal with kind of the limited sample size that you had, compared to other techniques? Because your sample size for the training testing isn't massive, right? So would you have been better off using something completely, unsupervised, like a PCA, for example, and looking at where people kind of fit into that, like higher dimensional space, or something a bit more basic?
Yeah, we didn't do PCA. I'm not sure. This data is I think it's some of it is older, so I think maybe that may have been attempted by somebody other than me in the past. But yeah, me and Jihan decided to try SVM, and the result was decent, so we moved forward with that basically. But there's there was still an effect of levodopa in reducing forelimb asymmetry in these animals. Yeah.
But they additionally had like a mobility disorder like symptoms.
Yeah. Yeah, they had hemi-Parkinsonism, which we measure basically. We measure how they're favoring one limb over the other. Using this test. Yeah.
Okay. And then you kind of like confirmed that also in like in your IHC that like there was something.
Yes, yeah, we confirmed with the IHC, and we also excluded animals without a full lesion, because that can because that can have a pretty big effect. And none of the animals without a full lesion, anecdotally, none of them develop dyskinesia symptoms. But it really is like the animals really only show symptoms after about like 70 to 80% of the cells are destroyed, right? So it's
Start treatment 35 days after injection. You don't like wait for symptom onset and then start the treatment, right? So you do the injection.
Yeah. We wait for the animals to recover, and then we do the cylinder test to confirm that the animals have Parkinsonism. But because that's not a super specific test, we also then do the immunohistochemistry to make sure. Yes.
All right. Run on time. There you go. Okay. And lots of good questions for Sam. Thank you very much.
Okay. Thank you very much. Thank you.